So in previous videos, we've discussed the three different energy pathways for resynthesizing ATP, just in general. Now let's focus on how ATP is resynthesized aerobically. Remember, this is the one it's the slowest process, but it's also the most sustainable, and it's forming the base of all of our ATP resynthesis. It's always running in the background. Your anaerobic processes you only use basically in case of emergency. So aerobic metabolism occurs in the mitochondria for the most part. Like you'll process like your fats, you'll start processing your fats and your carbohydrates outside of the mitochondria, but the ATP is ultimately resynthesized in the mitochondria. So I like these pictures. So you have your muscles, right? So you have your Z lines here. You can see in between the myofibrils and the sarcomeres, you have these mitochondria popping up. This is where your aerobic metabolism is going to occur. One of the key differences we see in people who are aerobic trained versus people who are not is how many mitochondria are stuffed in between their muscles. People who are aerobically trained, like endurance runners, will have a lot more mitochondria in there, a greater volume of mitochondria than someone who is not. Now, here's another look at at a muscle. So this is as if we cut the length of the muscle and we're looking down. So all these are like the dark little dots there are going to be your myosin and your actin are right around it. So we're seeing lots of little myofibrils right here. What this picture helps illustrate is that you have the mitochondria all over the place. And it's not just like one little bean, but it's more of a network or a reticulum. This is a kind of a funny lesson that you can learn from science. For years in most textbooks, and you'll even see me do this sometimes for simplicity, mitochondria are often illustrated looking something like that, like a bean with two layers in it. And the reason for that is because when scientists take the muscle or whatever they're analyzing to look at the mitochondria, they literally put the muscle in basically a blender, and it chops up the mitochondria, and then they form these little beans. And so we're just seeing partial mitochondria for a long time. It took for a while for the technology to get in place where we could see the mitochondria as they should look in the muscle. And we realized that it's a vast network or a reticulum of mitochondria, not just these isolated beans. Anyway, that's a tangent. So how is ATP resynthesized in the mitochondria? It's all about hydrogen gradients. Now, this is a chance where I get to take some of my Nevada pride. I'm from Las Vegas. And in Las Vegas, or right outside of it, we have the Hoover Dam, which has dammed up the Colorado River. And this dam is what provides electricity for a lot of Southern Nevada, Southern California, Arizona, and a lot of other places. And the way that dams make electricity is similar in concept to the way your mitochondria are going to make ATP or resynthesize ATP. It's all about gradients. And so you can see this dam has made a big gradient, right? We got a lot of water on this side of the dam and not a lot of water on the other. The dam is going to use that gradient to move a turbine. So over here, so here's the dam, a lot of water on this side. As the water goes down the gradient, it's spinning a turbine. And that turbine is what's generating the electricity that powers the Las Vegas Strip. So it's literally water going down a gradient, causing something to spin that makes the electricity. Now, your mitochondria work very similarly. So in your mitochondria, you have a couple of membranes. This is like the very outside of the membrane. So if I draw like the bean again, so this membrane would be right out there, and this one would be on the inside. Okay, so inside of the mitochondria, you have an enzyme called ATP synthase. And what ATP synthase does is it takes hydrogens from one side, an area of high concentration, and lets it flow down to an area of less concentration. So it's flowing down a gradient. The membrane acts as a dam, and the hydrogens go down across the dam, down the gradient. And as those hydrogens move down the gradient, it causes a spinning in part of this ATP synthase enzyme that is going to provide the energy to shove ADP and phosphate back together. So just like this turbine over here, we have something going down a gradient that causes something to spin and resynthesize ATP or provide us with our energy. So you might be looking at this and thinking, okay, so hydrogen ions are involved or protons are involved in making ATP. Where the heck does all the fat, the carbohydrate, where does the oxygen come in with this aerobic metabolism? I don't see it. So the whole point of burning fats and carbohydrates and consuming oxygen is to get the hydrogen ions up the gradient so that they can then go back down. Remember, it takes energy to put something against a gradient. These hydrogen ions aren't just naturally there. Under normal circumstances, without anything going on, you always get an equilibrium across membranes. And so we have to use energy to put all these hydrogens up here and so that it can then go down ATP synthase. And so that's where you get the fats, carbohydrates, and the oxygen consumption. It all facilitates the movement of these hydrogen ions to the intermembrane space. Now, this is where it gets a little more complex, but these four complexes, all they're doing is passing electrons that have been donated to them by fats and carbohydrates, essentially. They're passing them, and as they pass from one spot to another, they release energy. And as these electrons release energy, it provides enough energy to move hydrogen ions up the gradient. So in other words, if the electrons are being used to put water from one side to the other so that it can come back down. So the water can come back down and be used. Now, how does this work? So it all starts at complex 1, conveniently, of the electron transport chain. So inside of the inner membrane, you're going to have NADH. Now this NADH is going to be created or you're getting the NADH from glycolysis and from the citric acid cycle. So when you're breaking down these carbohydrates and fats, you take an NAD, you add electrons and hydrogens onto it. That's what the citric acid cycle is primarily for. And that NADH is going to come over here to complex 1 and donate its electrons. So the NADH donates its electrons and hydrogen or two and just becomes that empty NAD plus bucket where it doesn't have any extra electrons anymore. As the electrons go from complex 1 to something called coenzyme Q, which is a transporter, energy is released. And that energy release is what facilitates the movement of hydrogen up the gradient. And then electrons are passed from coenzyme Q to complex 3, from complex 3 to complex 4. And as it's moving from complex to complex, it's providing energy to move hydrogen up the gradient. Ultimately, the electrons are received by oxygen. Oxygen is known as the final electron acceptor. Without oxygen, these electrons have nowhere to go and they'll just stall. And you don't get any hydrogen ions moving and you won't get any ATP resynthesis. So the electrons ultimately bind to oxygen on complex 4 and form water. All the while, as the electrons move, they're moving hydrogen ions. So you also can donate electrons from FADH2. So this is also an electron carrier that's coming from the citric acid cycle and other places like beta oxidation. And FADH2 has these extra electrons that it just wants to donate. And it comes in at a slightly lower energy. And it donates them first to complex 2. And those electrons then make their way to complex 3 and then 4. So it bypasses complex 1. Now, what's the problem with that? Or what's the consequence of that? FADH2, when you're using or donating electrons through FADH2, you're skipping complex 1 and the 4 hydrogen ions that will be moved by complex 1. So you're going to move less hydrogen. And remember, the hydrogen is used to resynthesize ATP. So less ATP will be resynthesized via FADH2. Now, here comes a very important concept. Where does oxygen figure into ATP resynthesis? Is the oxygen becoming part of the ATP? Is it like used to like glue to hook the phosphate up to ADP? The answer is no. The oxygen does not become part of ATP. The oxygen, all it's doing is accepting these electrons so that hydrogen can move up. And then it's the hydrogen that moves down the gradient that provides the energy to put ADP and phosphate back together. This is called a coupled reaction. Oxygen consumption is indirectly linked to ATP production by way of the hydrogen ions. If the hydrogen ions found some other way to go down, let's say that there was another tunnel instead of ATP synthase, that these hydrogen ions could go down, you'd have lots of oxygen consumed to get the hydrogen up the gradient, but because it's bypassing ATP synthase, you'd get no ATP. So this is why it's coupled. It's not a direct conversion of oxygen to ATP. Oxygen just provides the movement of the hydrogen ions. So I already mentioned that FADH2 results in the movement of fewer hydrogens per oxygen consumed, right? All these 2 electrons are going to come down and bind onto oxygen. You're going to consume both FADH2 and NADH are going to consume 1 oxygen, but in the process, NADH will move 4 more hydrogens than FADH2. So the consequence of that is more hydrogens equals more ATP. So in terms of efficiency, if you had a choice, and you don't, but if you did, if you had a choice between only having NADH or FADH2, you'd probably want to go with NADH because you're going to get more ATP per oxygen. And it all comes down to moving those hydrogens. So now the question is, where does NADH and FADH2 come from? This is where we see food come into place, like glucose, carbohydrates, fatty acids. So here I've drawn a very simplified diagram of the electron transport chain and oxidative phosphorylation. Remember, we have NADH or FADH going in, donating electrons, and coming out as NAD or FAD. At the same time, we're consuming oxygen, but the main thing here is that we're going to get ATP out of all this process. So everything with your carbohydrates and fats is really, almost all of it is about getting NADH and FADH2. And so with a fatty acid, you're going to yield a lot of these things. So a fatty acid is a molecule that has a lot of carbons in it, a carbon chain. So each one of these little green things would be a carbon all the way through it. And these carbon chains can be short, like maybe 8, 10 carbons long. It can be over 20. Right? The way that you're getting NADH and FADH2 out of these fatty acids, or by burning these fatty acids, comes down to a couple of steps here. So let's say you have a fatty acid, make it into your muscle cell. That fatty acid, the first thing you're going to do is break it down into pieces. You're going to cut it in 2 chunk pieces. And this is called beta oxidation, where you're cutting the 2 carbons off at a time. And for every cut that you make, it's going to yield 1 2-carbon chain, which we'll use in a second, but every cut also yields an NADH and an FADH2. And those are then going to go down to the mitochondria to be utilized in oxidative phosphorylation. So just splitting up, ripping the carbons off of the fatty acid provides us with energy and electrons to make FADH2, and NADH that go to the electron transport chain. But it also provides us with acetyl-CoA, which has some energy in it as well. And that acetyl-CoA is going to go to something called the citric acid cycle. And acetyl-CoA comes in and binds with a 4-carbon molecule to become a 6-carbon molecule, and it's going to go through this cycle and eventually kick off another carbon to go back down to 5, and then kick off another carbon to go back down to 4. And in that process of going from 2 to 6 to 5 to 4 again, you're yielding some NADH and FADH2. You'll yield 3 NADH for every acetyl-CoA that comes in and an FADH2. In the process, you'll also yield an ATP and some carbon dioxide. The ATP is actually indirect. It first becomes something called GTP, which is quickly converted to ATP. So in terms of carbon dioxide, when you're exercising, you take in oxygen and you exhale CO2. It's not that we're converting oxygen into CO2, right? What are we converting oxygen into? In the electron transport chain, we convert the oxygen into water. And then in the citric acid cycle, as we're kicking off these extra carbons, we make a CO2, a carbon dioxide. So this is where they're coming from in fats. But what about glucose? So glucose is a 6-carbon chain, not as many carbons as a fat. But we're going to process glucose through something called glycolysis. We're going to take it from a single 6-carbon chain to 2 3-carbon chains. And in that process, which you'll learn in a couple of days, you'll yield some ATP and also some NADH and FADH2. I'm missing something here. You also have an NADH come off of here during glycolysis, which will go down here. But then that pyruvate, these pyruvates are these 2 3-carbon chains, will then be converted to an acetyl-CoA or a 2-carbon chain. In that process, they kick off a carbon, which becomes carbon dioxide, another source of your CO2 when you exhale. And then that acetyl-CoA enters into the citric acid cycle. So acetyl-CoA is kind of like the midpoint, or where the metabolism of fat and carbohydrate meet at acetyl-CoA. But this is where our NADHs and FADH2s are coming from. Citric acid cycle will give you a little bit of ATP, but it's far more lucrative to get the NADHs and the FADH2 out of it. Remember, every NADH is going to give you about 3 ATP. Every FADH2 is going to give you about 2 ATP. So let's zoom in on the citric acid cycle. So here's the citric acid cycle. Let's zoom in and see what it looks like. We have that 3-carbon pyruvate that comes in, and then you're going to rip off one of these carbons, and it becomes a CO2. Now you have this 2-carbon acetyl-CoA. That 2-carbon acetyl-CoA is going to hook up with oxaloacetate, which is a 4-carbon chain. So 4 plus 2 equals 6. So that becomes citrate, which is where the idea of citric acid comes from. So we've taken a 2-carbon acetyl-CoA and a 4-carbon oxaloacetate to make a citrate. And then through several steps, we're then going to just gradually rip off these extra carbons, the 6th one and the 5th one, and it'll eventually go back to being oxaloacetate to start the process over. But why would we do that? Why would we put on carbons just to rip them off again? Well, the idea of doing that is as you rip off this first carbon, it provides an electron for NAD plus, so that you now get an NADH. And that CO2 you exhale, that carbon that you've ripped off, becomes that CO2 there. And now you're down to 5 carbons. Is that 5? That's 5. So now you have 5 carbons, and it's going to go through a couple more steps where you're using enzymes to process and move the carbons around, move hydrogens and electrons and all these other things. And as you do that, it releases enough energy to make a GTP and ATP. But then you're also going to move around enough that will provide an electron to FAD. So now you got FADH that's going to go down to the electron transport chain. Now, sometimes in books you might see it listed as FADH, FADH2. Sometimes you'll see NADH plus H plus. I'm not going to get too technical on this. If you see an H on the NAD, that means it has an extra electron. Just assume it's full. If you see an H on the FAD, assume that it's full of electrons. If you don't see an H, assume it's empty of extra electrons. All right, so by breaking down this 5-carbon glucose, we've made an FADH2 that's going to go to the electron transport chain and provide us with the movement of hydrogen ions. And then we're going to keep on going, and we'll convert again, moving around these carbons and these hydrogens, and we're going to yield another NADH. So I think I got a little out of order here. I don't care that you know exactly when carbons are coming off and which enzymes are doing it. But if you get into another class, they might care a little bit more. So the first carbon is coming off at this point in the cycle, and the second carbon is coming off there. So once we get to succinyl-CoA, we're down to a 4-carbon chain again. And then it's just rearranging these carbons and other hydrogens and things that yields the rest of the energy. So that's a lot of talking just to say that by pushing carbons on and off of oxaloacetate and citrate, we can yield NADH and FADH2 that can then go to the electron transport chain. I do not expect you to memorize all of these enzymes and all of these steps. Do remember how many NADH and how many FADHs are going to come out of the citric acid cycle. Also remember how much CO2 is produced and ATP or GTP. But don't spend your days memorizing that fumarase takes fumarate to malate or anything like that. You got better things to study. All right, so we've made all these NADHs, right? And these NADHs and FADH2s from the electron or from the citric acid cycle and glycolysis and beta oxidation, they're donating the electrons that then move down a gradient or move from high energy to low energy. Eventually they bind to oxygen, and while they do that, all those move hydrogens, and the hydrogens go down your gradient. And as they go down the gradient, it resynthesizes ATP. Kind of talked about this already. And this is just an illustration. You can pause this and look at it a little bit more. This is another way of looking at beta oxidation of fats in a little more detail. I don't expect you to be able to look at like a 16 carbon chain and say, oh, 129 ATP come out of it. What I do expect you to understand is that a lot of fat or a lot of ATP can be yielded by burning fat. And then in terms of glucose, it's about 32 to 38. It depends on the efficiency of the mitochondria and several other things for exactly how much are going to be netted or gained by glucose. But just remember, it's about 34 to 38. I'm not going to ask you specifics. All right, so this process I referred to earlier of oxygen consumption is indirectly linked to ATP consumption, right? Its oxygen consumption is linked to it by the oxygen consumption allows for the movement of the hydrogen ions, and then hydrogen ions going back down the gradient is what actually resynthesizes the ATP. So this is what I'm called a coupled reaction. But you can uncouple this. What happens if we bypass this? So let's say that we're consuming all this oxygen to move these 10 hydrogens up into the intermembrane space. And that as these hydrogens go down the gradient, they're going to resynthesize ATP. It's somewhere around, estimates are like for every 3 hydrogens, you get about an ATP. Just for simplicity, let's say that for 10 hydrogens, all of these, you'll get 1 ATP. That's not really what it is, but just simplicity. So you have to move 10 hydrogens to get a single ATP. 10 hydrogens have to go down ATP synthase to get an ATP. But what happens if some of them go down a different way? You have these proteins called uncoupling proteins that allow the hydrogen ions to go down the gradient in ways besides ATP synthase. This would be like a leak in your dam. It's going down the gradient in a way that you can't harvest energy from it. So what that means is if you have some kind of inefficient setup here where the hydrogen ions are going down the gradient another way, you're going to have to consume more oxygen, burn more fats, burn more carbohydrates to get more hydrogen ions up here to get the same amount of ATP. This is to illustrate that it's a coupled reaction. Your oxygen consumption and your fat burning and your carbohydrate burning do not directly lead to ATP. They lead to hydrogen ions, and hydrogen ions going down ATP synthase lead to ATP. If the hydrogen ions that you burnt all your fat for and consumed all the oxygen for, if those go down some other way besides ATP synthase, you don't get ATP for it. Now, you might be like, well, why on earth would you have that? There are reasons for it, and everybody has it. It's in every cell. It has a little bit of this uncoupling or this inefficiency. Some of it is due to these proteins, which are called uncoupling proteins. You have other channels called ANT channels that allow the hydrogen ions to go down. Also, there's some theories that some of the hydrogens might just be able to slip down the membrane, like especially in older damaged mitochondria, you might conceivably have holes in this membrane that allow the oxygen or the hydrogens to go down the gradient and not get ATP for it. Either way, it makes you very inefficient, right? It makes you have to burn more calories to get the same amount of ATP. And so uncoupling proteins, it's a lot like letting water go down the gradient of a dam, not through the turbine, right? We only get electricity from Hoover Dam if the water goes past that turbine. If it goes some other way, like through a hole in the dam or through like an emergency outlet valve, you don't get electricity for it. Same thing. If you have hydrogen ions going down the gradient, not through ATP synthase, you don't get ATP for it. So why would you use this? There are a lot of theories on what it can be for. One is for heat generation. You see this a lot in babies. Babies tend to have fat. The mitochondria in their fat tends to have a lot more of this uncoupling protein. And what that means is that they have to burn more fat, emphasis on the burn here. They have to burn more fat to get the same amount of ATP. So they're producing more heat at rest, and it helps keep them warm. So you can either look at metabolism as the purpose of metabolism being to produce ATP, or you could alternatively look at it as the purpose of metabolism is to produce heat. If you actually go by what's produced more, it's heat. We're only about 32% efficient. So 32% of the calories that we're burning are used to make ATP. The rest of them, 68% roughly, go off as heat. So really, we're making a lot of heat, and we just happen to make ATP in the background. At least that's what's going on in these babies with what we call brown fat that have a lot of these mitochondria in it that have uncoupling proteins. They're very inefficient, and so to meet their resting ATP demands, they have to burn more fat and produce more heat, and it just helps them stay warm. Other things that can influence this, cold exposure is thought to potentially impact the expression of these uncoupling proteins so that your resting metabolic rate increases, you burn more calories, produce more heat just to have the same amount of ATP to live. There are other things that you might be able to take that decrease uncoupling. One of them right now that's kind of a topic of interest in exercise physiology is beetroot juice or nitrate supplementation. And the nitrate supplementation taking in beetroot juice or drugs that have nitrate in it seems to decrease the expression of uncoupling proteins and other similar proteins, so it makes you a little more efficient. So you get more ATP per oxygen. Thyroid hormone is also thought to impact this uncoupling. All right, so the take-home message on aerobic metabolism, it's a coupled reaction. The aerobic refers to the oxygen consumption, and that oxygen consumption is used to get the hydrogens up there. The ATP resynthesis comes when those hydrogens go down ATP synthase and spin part of that enzyme that basically squeezes ADP, an inorganic phosphate, back together. Where do carbohydrates and fats come in? Well, those are our sources of NADH and FADH2. We're processing those and burning them so that we can get these NADs filled up with electrons to become NADH to provide electrons to the electron transport chain. Make sure you can explain this, and make sure you can explain what uncoupling is. If you understand what uncoupling is, it helps you really grasp how all of the electron transport chain and oxidative phosphorylation are working. All right, that's it. I hope you guys have a wonderful day. I'll talk to you later.